This method can help answer key questions in the DNA repair field, such as how DNA repair complex formation, localization, and resolution are regulated. The main advantage of this technique is that it can unambiguously detect changes in repair complex localization and kinetics.
In addition to these undergraduate researchers in my lab, 3 graduate students will perform the experiments. That will be Changkun Hu, Dalton Dacus, and Stephen Walterhouse.
To begin, grow U2OS-DRG-FP cells on a 10 cm tissue culture plate until they are 85 to 90% confluent. Then remove the medium at 3 ml of EDTA and incubate the cells at room temperature for 3 minutes. Replace the EDTA with 1 ml of trypsin and incubate the cells at 37 degrees Celsius for 5 minutes. Then add 5 ml of DMEM to neutralize the trypsin. After counting the cells, seed 6 times 10 to the third cells per well in 200 microliters of medium in a glass bottom 96 well plate. Alternatively, seed 4 times 10 to the 6 wells in 8 milliliters of medium on 12 mm cover slips placed in a 10 cm plate. Then grow the cells overnight at 37 degrees Celsius and 5% carbon dioxide. To induce double-strand breaks, or DSBs, replace the medium with hydrogen peroxide diluted to a concentration of 25 micromolar with DMEM plus 10% FBS and incubate the plate at 37 degrees Celsius for 1 hour. Wash the cells 3 times with 1x PBS, then add fresh DMEM. To fix the cell,